Okay, so we have finished the introduction to the enzymes. Now, the second topic that we have to take is the classification of enzymes. Why do we want to learn the classification? Because it's always a big job for everybody to learn the name of enzymes, especially if you're cramming the name of the enzyme. You will be like out of the world if you cannot learn the name of all the enzymes. But if we classify and learn, understanding the reaction mechanism, and then learning the name of enzyme, it will be a very easy job. Maybe 80 or 90 percentage of the enzyme's name you can just learn by seeing what is the reaction. Okay, so that is possible only if we learn the classification of enzymes. And why do we want to learn the name of the enzyme? It is because whenever an applied question is coming, the question usually what is the enzyme defect. So if you want to know the enzyme defect, you should be knowing what is the reaction. So only if we know the reaction, we can know the name of the enzyme easily. Otherwise, we'll just by heart everything. So in order to avoid all these things, we should be knowing about the classification of enzymes. Okay, so if suppose like if you are joining a medical college, suppose there is one more. What's your name? Okay, whatever be, you have a name. So in your name itself, there is two more are there which has joined the same medical college. For example, in my name is Rebecca James. I have like when I joined college, there are two Rebecca James in my class. Okay, so if the teacher is calling Rebecca James, both these people will stand up, right? So this will make a big chaos in the calculation of marks, in assigning like internal assessment, your attendance, everything. There will be problem. Okay, so in order to prevent that, what we'll do, we will do a unique name or a unique roll number for every person. For example, based on the date of birth or the order, the admission number, etc., we are going to give a unique number to every person. For example, first Rebecca James, who has like a first join, will be that roll number first will be given to her, and the second person will be given the second number, like that. So this is what happens in our like in the roles. Similarly, in the case of enzymes, for example, if I say galactosidase. Galactosidase is an enzyme which can remove galactose, but actually this galactose can be removed from various substrates. So based on that, these two reaction where it is removal of galactose is same for these two enzymes, but actually they are acting on two different substrates, so they are two different enzymes. But trivially, we are giving the same name, galactosidase. Like that, there are so many enzymes which are named after their reaction mechanism. They will be having, so if two enzymes are having the same reaction mechanism, we will be giving the same name. It creates confusion, because which is that enzyme will always be a confusion. So that is what is called as trivial name based on reaction. Mostly it is based on the reaction mechanism. Sometimes it can be based on the substrate it acts, like that. So most of them, it is based on the reaction mechanism, the trivial name. So that is one thing. But so in order to prevent such chaos, what we do is that we are giving a unique number to every enzyme. So that was done by an international union of biochemistry and molecular biology, that is called IUBMB. So IUBMB give a unique number to every enzyme. This is called enzyme commission number or enzyme class number, or it is called enzyme code number. Okay, so this enzyme code number has 4 digits. So let this be A, B, C, D. Even though I have written alphabets here, it is not alphabet that we are giving. We are giving a unique number. For example, 1, 1, 1, 1. Okay, so I'll take this number 1, 1, 1 here. Okay, this is standing for an enzyme called as alcohol dehydrogenase. So what is this first digit standing for? The first digit is standing for the class of the enzyme. Okay. The second digit is standing for the subclass. Okay, the third digit is standing for the subclass. And there is a fourth digit which is a unique number for every enzyme. In that subclass, a unique number is given to each and every enzyme. So there are 4 digits in enzyme classification or enzyme code number. So this is the sequence. Okay, fine. So once that is clear, we are just concentrating on this enzyme class, and subclasses in only a certain class I will be describing the subclass. Apart from that, we are not going to learn all the subclasses. So we are going to learn this first digit, which is like numbered from 1 to 7. In other words, what we can understand is that there are the first digit, there is 7 in number. That is, it means that there are 7 classes of enzymes numbered from 1 to 7. Okay, fine. Now we are going to see which are these classes of enzyme. So to learn the classes of enzyme, I will give a simple mnemonic for you. Operation, theater, has, low, intensity. This T I am writing capital. Okay. So O is one class, T is standing for another, H is standing for another, L, I, L and T. So these are the first alphabets of every class. So what is that O standing for there? O stands for oxidoreductase. And the T stands for transferase, which is class 2. Then class 3 is hydrolase. Then the fourth class is a lyase. Then the fifth class is an isomerase. And that L again, that is a ligase. So in the previous, you can see that LIG here, right? So that stands for ligase because there are 2 L here. The first L is lyase. The second LI, it is LIG. So it is LIGASE, ligase. And we have a last T here. And that T is translocase. This is a recently added one in August 2018 onwards. This last 7th class is added. Okay. Now we are going to see what are the examples of each of these classes, starting with oxidoreductases. So oxidoreductases is class 1. Remember, we cannot put a class 1 enzyme to class 2. So class 1 enzyme should always be class 1. Class 2 enzyme should always be class 2, like that. So class 1 is oxidoreductase. The name itself is telling you what are oxidoreductases. Oxidoreductases are enzymes that catalyze oxidation, reduction, reactions. Fine. Now, under this oxidoreductases, we learn the subclasses. The subclass number 1 is dehydrogenase. What are dehydrogenases? Dehydrogenases, the name itself is telling you what is it. It is catalyzing the transfer of hydrogen elements like H plus, H minus and H2 and also electrons. So all the hydrogen elements are transferred to an acceptor in a coupled oxidation reduction reaction. So whenever hydrogen and electrons are involved or oxygen is involved, we know that it is an oxidation reduction reaction. Specifically in dehydrogenase, it is removal of hydrogen elements. Okay, and it is given to an acceptor. So what is the acceptor here? The acceptor here is usually NAD plus, or it can be NADP plus. Both these, if NAD or NADP plus is there, we call it as a nicotinamide. Nicotinamide enzymes. And if FAD, this FAD also can be the hydrogen element acceptor, then it is a flavoprotein. Flavoprotein. Okay. So usually the acceptor for this hydrogen elements can be nicotinamide derivative like NAD or NADP plus, or it can be FAD. So in dehydrogenases, what we can learn is, in general, any enzyme with dehydrogenase in its name, any enzyme with the dehydrogenase in its name, every time you can write the coenzyme, because it is a vitamin that is acting as, it's an organic molecule that is acting as the second substrate or a co-substrate or a non-protein part. So that's a coenzyme, is usually NAD plus, then it will become NADH, or it can be FAD to FADH2. And the third thing is that it can be NADP plus to NADPH. So usually these are the hydrogen element acceptors. NADPH. Okay. Now, which are the examples under this? It is difficult to learn the set of enzymes where NAD to NADH is there, because there are lots and lots of enzymes in our body where NAD is converted to NADH. So it is difficult. So we can learn something which is very less in number, and whatever others are there will be NAD to NADH. That is the easy way to learn, right? So we will learn which are the enzymes where FAD is converted to FADH2. So that is that we can confine only to 3 in number, FAD to FADH2. The first one being acyl coenzyme A dehydrogenase, which is an enzyme in the beta oxidation. The second is succinate dehydrogenase, which is an enzyme in the TCA cycle. Then the third one is glycerol 3-phosphate dehydrogenase, and specifically this is mitochondrial. Cytoplasmic is also there, it is not FAD to FADH2. So these are the only 3 enzymes where FAD to FADH2 is there. So we can learn that. Then comes the NADP to NADPH. This is also very, very important set of enzymes where these are called as NADPH generators. Okay, so that is also very less in number. Like, one is the hexose monophosphate pathway, HMP shunt pathway, where 2 enzymes, the first 2 enzymes in the HMP shunt pathway. I'll just write the names here. It will be difficult for you to learn now, but still I'm writing it. One is the glucose 6-phosphate dehydrogenase, okay, G6PD. Then the second is 6-phosphogluconate dehydrogenase. Then cytoplasmic isocitrate dehydrogenase and malic enzyme. So these are the NADPH generators. It is very, very important. NADP is converted to NADPH. Only these 3 enzymes are involved, which means that whatever other dehydrogenases we are writing in any metabolic pathway, usually it will be NAD to NADH, or it can be in the vice versa. If it's a reversible reaction, it can be in the other direction also. So generally, I'm saying NAD to NAD plus to NADH. Okay, so these are the just learn that in dehydrogenases it is a removal of a hydrogen elements, and usually whenever we write dehydrogenase in the name of the enzymes, always there is a coenzyme that we have to write along with it, either FAD to FADH2, NADP to NADPH, or NAD to NAD plus to NADH. NAD to NADH is very difficult to learn. The rest I've already given you the list of enzymes. Okay. So after dehydrogenase, under the subclass of oxidoreductases, we have the second one under oxidoreductases, it is oxidase. Oxidase means in the dehydrogenases, it is removal of hydrogen elements, but in oxidase, it is removal of hydrogen from the substrate, and it is given to an acceptor. In the case of dehydrogenase, it is given to an acceptor which is usually a B complex vitamin. But here the difference is that hydrogen elements are removed and it is given to an acceptor. And who is the acceptor here? This makes the difference here. Oxygen is the hydrogen acceptor here. That is the difference here. So Whenever oxygen is the hydrogen acceptor, it can form two products. Either it can be an H2O or it can produce H2O2 as well. So this is very important because it is an H2O2 generator. So which are the examples under this? The examples under this are cytochrome C oxidase. Cytochrome C oxidase is the complex 4 of electron transport chain. If you remember, if you are knowing what is an electron transport chain, you know that oxygen is the final electron acceptor which gets reduced itself to water. So it accepts a hydrogen element along with an electron, and finally it is getting converted, it is getting reduced to water. So same definition we can fit into. The other is the monoamino oxidase. Then comes the L-amino acid oxidase, another example of oxidase. Then one more is there, it is xanthine oxidase. So these two usually produces hydrogen peroxide. Okay, so definitely it is a transfer of a hydrogen element, but usually the acceptor of this hydrogen element is oxygen. That is called as oxidase. Then what is oxygenase? Again, it is a subclass of oxidoreductase. So we have learned dehydrogenase, second we learn oxidase, now it is oxygenase. Oxygenase is like it adds oxygen directly to the substrate. It is it. Add the oxygen directly to the substrate. So based on that, it is divided into two. So if it add one atom of oxygen, or it can add both atoms of oxygen. So if it is adding only one atom of oxygen, it is called monooxygenase. And if it is adding both atom, it is called dioxygenase. Okay. Monooxygenases, example, phenylalanine hydroxylase, then tyrosine hydroxylase, tryptophan hydroxylase. All these are example, plus 7-alpha hydroxylase. And most of the cytochromes, they are monooxygenase. There are other names for this monooxygenase. It is also called as mixed function oxidase, or it is also called as, most of the monooxygenases are hydroxylases. So you can see here, these are all hydroxylases. So in general, what we can say is that most, not every, most hydroxylases are monooxygenase. Okay, fine. So these are the examples of monooxygenase. And remember, phenylalanine, tyrosine, tryptophan, all these are what? They are aromatic amino acids, right? So we can learn that all aromatic amino acid hydroxylases are monooxygenase. Fine. So these are the examples of monooxygenase. Now going to the dioxygenases. Now we have to learn about what is called as dioxygenase. Dioxygenase, it adds both atoms of oxygen that we have already seen. Next is the example. The examples are homogentisate dioxygenase, then tryptophan dioxygenase. So these are examples of dioxygenase. So we have learned about what are called as oxygenases. Now we are left with what is called as hydroperoxidases. So first, the hydroperoxidases, there are two enzymes, or there are two subclasses under the name hydroperoxidases. One subclass is peroxidase, the other subclass is catalase. So this is all coming under oxidoreductase. So under oxidoreductases, we have learned dehydrogenases. We have learned, second, we have learned what are called as oxidase. Then we have learned what are called as oxygenase. Then the next one is the hydroperoxidases. Hydroperoxidases have two numbers under that. Under the name of subclass called as hydroperoxidases, there are two types of enzymes. One is the peroxidase, the other is catalase. What is common to these? The common thing for these two is that they use hydrogen peroxide or an organic peroxide as its substrate. It is common for these two. Usually they use hydrogen peroxide as its substrate. That is common. Both use hydrogen peroxide. Now, what is the difference here? The difference between the peroxidase and catalase, what is the acceptor of electron here? There comes the difference. So in the case of peroxidases, the electron acceptor, so there comes the difference. The electron acceptor can be glutathione or vitamin C or quinones, cytochrome C. So these all can be the electron acceptor. I will give you one example here. That is hydrogen peroxide is converted to water, and here you can see glutathione. So glutathione, we write it as GSS. You will be learning about this later also. GSH is glutathione. It is converted to oxidized glutathione. One more oxidized glutathione come here, so that we write it as oxidized glutathione. So this is oxidized, this is reduced glutathione. Okay, fine. So hydrogen peroxide is converted to H2O2, where this glutathione is acting as the electron acceptor, and the substrate here is hydrogen peroxide. Okay. Now this enzyme is coming under peroxidase, and that enzyme is glutathione peroxidase. Okay, so this is an example of peroxidase. Now comes the catalase. Now catalase itself is the example. What is the difference in catalase? In the case of catalase, hydrogen peroxide is the acceptor as well as the donor. So H2O2 we have to write 2. One is the acceptor, one is the substrate, the other is the acceptor of the electron, where you can see that H2O2 is converted to 2H2O, 2H2O, 2H2O plus O2. So this is the reaction of a catalase. There are no other examples. The catalase itself is only one example here. So these are all coming under hydroperoxidases, where hydrogen peroxide is the acceptor, the substrate, but acceptor can be different. One, it can be any other acceptors like glutathione, ascorbate, as given you examples. The second is that H2O2 itself is the acceptor. Okay, fine. So these are the various subclasses under the heading oxidoreductases. So now we are moving on to the class 2 enzyme, which are called as transferases. What are transferases? The name itself is telling you something, right? It is the transfer of a functional group. Now, this functional group need not be a hydrogen element. It can be any other functional group other than hydrogen element, because if hydrogen elements are involved, then it will be a oxidation reduction reaction. So if the transfer is for any other functional groups, apart from the hydrogen element, we call it as a transferase. Okay. Now, what are the examples here? The simple examples are any enzyme with trans in its name. For example, transaminase, transketolase. Lots and lots are there. So any enzyme with trans in its name. The second example is, I am writing the reaction. Glucose is converted to glucose 6-phosphate. Now in this reaction, 1 ATP is converted to 1 ADP. In this reaction, what happens is a phosphate is transferred from this ATP to the glucose, so that the six stands for the sixth position. We know that glucose has 6 carbon atoms. So the sixth position of the glucose is getting phosphorylated using ATP as the donor. Now, so this transfer of phosphate is by an enzyme. Whenever transfer of phosphate is involved, we call that enzyme kinase. Okay. Now, in this case, this glucose can be, it can be galactose. So, it can be any hexose. Okay. So, this name is, we write either as hexokinase. If specifically for glucose only, we can also write it as glucokinase. Let it be anything. We just learned what is it. That is transfer of phosphate from an organic molecule which is called as ATP. Then we call it as kinase enzyme, and kinase belongs to which class? Class 2. Other example, the classical example is fructose 6-phosphate is converted to fructose 1,6-bisphosphate. Now in this reaction also what we can see is 1 ATP is donating the phosphate group, so that 1 ADP is formed. Now, whenever that ATP is acting as a donor, we have to write it as kinase, and this kinase is called phospho, because there is a phospho here. Then fructo, because there is a fructo here. So this is called phosphofructokinase. We write the short form here, that is PFK. Okay, so these are examples of kinase. So what we have to learn here is all kinases belongs to the class of transferases. So kinases are transferase. Then I'll give you the next example. That is, what we can see is glycogen. We know glycogen is a polymer of glucose. So let that polymer contain n number of glucose. This glycogen will give off one glucose. Let it give one glucose, so that now the number of glucose becomes n minus 1. So n is the number of glucose. When this glucose is given off, it is not given like that. It is like this glucose. This glucose is given to a PI, or an PI means inorganic phosphate. Inorganic phosphate. ATP is an organic phosphate. This is an inorganic phosphate. So that this PI is becoming glucose 1-phosphate. Okay. So what is the reaction happening here? It is like an inorganic phosphate is added to the glucose that is cut from the glycogen, so that the glucose is becoming glucose 1-phosphate. But this is not a kinase, because in kinase it's always an ATP involved. But here there is no ATP involved. So this is not a kinase. This enzyme, we call it as phosphorylase. So this phosphorylase is glycogen phosphorylase. So what we have to ultimately learn from there is this phosphorylases are transferases. Okay, so we have learned about the transferases. Any enzyme with the trans in its name, all kinases are transferases, and the third thing is phosphorylase is also a transferase. So the class 2 is over. Now we are moving on to the class 3. Class 3 is operation theater has. Now we have reached the H. So H is hydrolase. The name itself is telling you what is it. It is hydro and then lase, which means it is a hydrolytic, because it is like a breaking of a covalent bond. It's a cleavage of a covalent bonds like CC, CN, all these bonds are broken, and when these bonds are broken, a water is involved in that breaking up step. So that is called as hydrolase. So it is cutting with the help of water. So it is a hydrolytic cleavage. Okay, fine. Now, what are they cutting? They are cutting some covalent bonds. Where do we find the covalent bonds? Covalent bonds are usually found in the primary structure of carbohydrates, lipids, nucleic acid, etc. So these are all the complex biomolecules. We know that. So these biomolecules, it is like it is having this covalent bonds. Okay. Now, whenever we want to break this covalent bond, and if a water molecule is involved in this breaking, we call these enzymes hydrolase. Okay. So we have written the list of molecules which is having these covalent bonds right from the carbohydrates. The which is the covalent bond present in carbohydrate? It is the glycosidic linkage, right? Now, what about protein? The protein has peptide bond, right? Nucleic acid, it has a 3 prime to 5 prime phosphodiester bond. What about lipids? It is an ester bond. Okay, so these are all covalent bonds. Now, when we are adding a water and breaking this molecule, now the enzyme that do this will be coming under which class? Class 3. Class 3 is hydrolase. Okay, now we are going to learn the examples of hydrolase. So breaking of these carbohydrates, we know that the enzymes are amylase, then maltase, lactase, sucrase. So all these are breaking a covalent bond by adding water. Next is the peptide bond. Peptide bond is broken by protease, peptidase. Okay. Now, then under the proteases, we know there are various types of proteases like trypsin, chymotrypsin, elastase. Then we all know that pepsin. All these are proteases. So all these are proteases, or all these are breaking the peptide bond, and they all belong to which class of enzyme? Hydrolase. Fine. Then the nucleic acid, it is broken by exonucleases. Exonucleases. Then ester bond. It is by lipases or esterases. Okay. So what we are ultimately learning from here is all these enzymes are hydrolase. Okay. Now, what is the common term that we can call for all these enzymes? All these are digestive enzymes. All these are digestive enzymes. Fine. So all digestive enzymes, in a simple way, even if I've made you write a lot of things, it's like very simple to learn. Just learn one sentence here. That is, all digestive enzymes are which class? Hydrolase. Okay. Now, what are the other examples? Other examples I will write here. Glucose 6 phosphate is converted to glucose. Or in other words, one more reaction, fructose 1,6 bisphosphate is converted to fructose 6 phosphate. In all these reactions, what we can see is a water is added and a phosphate is released. It is not released as an ATP, it is released as an inorganic phosphate which is called PI. Okay. So water is added, a phosphate is removed. So it's a breaking of a covalent bond by adding water. Now this enzyme we call as phosphatase. Both these are phosphatase. Okay, what phosphatase? Write the name of the substrate here, glucose 6 phosphatase. Now this is fructose 1,6 bisphosphatase. Okay, fine. So all these phosphatases, what or which class they belong to? Phosphatase belong to, that is what we have to learn, belong to class 3, hydrolase. Okay, then one more example we can learn here is, as an extra point, is arginase. Arginase is an enzyme in the urea cycle. So in there we will be learning more. That is, a water is added, urea is released. So this is also a hydrolase. So in the hydrolase, what we have learned is all digestive enzyme, all phosphatases and arginase. These are examples of class 3 hydrolase. Now we are going to class 4, lyase enzyme. It is a tricky class where it is not having a single definition. First definition is very simple. That is, in the case of lyase, we don't add hydro in the name, which means that a water is not involved in this breaking. Okay, so it is like cleaving a covalent bond like CC, CN, CN, CO, etc., just like what we have learned in the hydrolase, without adding water. So for that example, it is one common example that we can learn is the aldolase. So aldolase. Aldolase is an enzyme which break a 6 carbon fructose 6 bisphosphate. This is a 6 carbon compound. It is cut into two pieces so that the dihydroxyacetone phosphate gets the first, second and the third carbon atom. Glyceraldehyde 3 phosphate got the rest of the carbon atom. So it is just like cutting the covalent bond without adding water. So that is fitting into the first definition, but not all. The second definition is it's by atom elimination, it forms a double bond. By atom elimination, it forms a double bond. I will give the example here. This is the classical example. This is the example of one of the enzymes in the glycolytic enzyme, which is glycolysis, which is the enolase. Okay. Now, in the enolase in the enolase reaction, what we can see is this 2-phosphoglycerate, which is a 3-carbon compound, is like it is not broken because the product is also 3 carbon. Okay, this is 3 carbon, this is also 3 carbon. So it is not like that. Then what is the difference here? You can see that this is the first carbon atom, this is the second carbon atom, and this is the third carbon atom here. Okay. Now to the second carbon atom, you can see a phosphate group. That is why it is called 2-phosphoglycerate. Now when enolase is acting, what is the reaction that is happening is an elimination of certain atoms where a water is eliminated. Once the water is eliminated from here, what we can see, this is the first carbon atom, this is the second carbon atom, and this is the third carbon atom. So between the second and the third carbon atom, what we can see is a double bond is created. So it forms a double bond by atom elimination. Similarly, this is a reversible reaction. In the other direction, it will add a water molecule and this double bond is again converted back to a single bond, right? So both reactions, as it is reversible, it can happen in either way. So in one direction, it is atom elimination. In the other direction, it is atom addition. But still, in the definition, we will say only atom elimination. Now again we see that, so this is atom elimination forms double bond. Then another definition is, the third part of this definition is it add groups across the double bond. What does it? Add groups across the double bond. We'll see the example here. The example here is the example of fumarase. This is a reaction in the TCA cycle. So in that reaction, what we can see is there is an enzyme, there is a substrate called fumarate is converted to malate. Okay, now when this reaction is taking place, what we can see is H2O is added, water is added, and this is the first carbon atom, second, third, and fourth carbon atom. Okay, now once the reaction has occurred, what we can see is this is the first, second, third, and fourth carbon atom. So here, water, you can see that NOH, this was a double bond. Between 2 and 3, there was a double bond. A water is added, and in this, when water is added, you can see this is the NOH that is added across the double bond. So between the 2 and 3, NOH is added. Again, one hydrogen is also added. An H and OH is added across the double bond, so that the double bond has now become a single bond, which is a reaction that is called as fumarase, which means that it is like an adding molecules across, or adding atoms across the double bond so that the double bond is now becoming a single bond. So these are all coming under lyase. So we have learned under lyase the examples aldolase, then enolase, then we have learned fumarase. Apart from that, you learn one more thing, that is all enzymes with lyase in its name. Okay, so aldolase, fumarase, enolase, and all enzyme with lyase in its name are the class 4, which is lyase. The enzymes with lyase in its name, example, if I'm saying HMG coenzyme lyase, arginino succinate lyase, these are all examples. Okay, fine. So these are all coming under the lyase. Then we have other examples also, because it's conceptually important. That's why I'm saying it here. The fifth example of a lyase is decarboxylases. I'll give you some examples here. Histidine is converted to histamine, where a carbon dioxide is removed. So it is atom elimination, histamine. Then if it is tryptophan, it will become tryptamine. If it is tyrosine, it will become tyramine. So if it is glutamate, tell me what it is. So histidine to histamine, tryptophan to tryptamine, tyrosine to tyramine, what will be glutamate? Is it glutamine? No, it is not glutamine. It is gamma aminobutyric. That is GABA. So don't forget it. Then the fifth example is dihydroxyphenylalanine becoming dopamine. From dopamine, we should get epinephrine and norepinephrine. Why I am saying all these things? It's for a reason. For all these decarboxylation, we need a vitamin. And that vitamin is PLP. PLP, that is vitamin B6. So, there is a clinical condition called as pyridoxine dependent seizures, where a neonate presented with intractable seizures, not responding to any anticonvulsant, but it responded well to vitamin B6. So they are called pyridoxine dependent seizures. Why? What is the reason for this, biochemical reason for this pyridoxine dependent seizures? It is like, GABA. What is it? It is a neurotransmitter. Dopamine, again another neurotransmitter. Epinephrine, norepinephrine, all these are catecholamines. So they are all neurotransmitters, which means if there is a B6 deficiency, there is a gross deficiency of all these neurotransmitters, which will affect the brain, which lead to seizure. So all these reactions are, we call it as simple decarboxylation. It is belonging to the class of lyase. And all these require B6 acids coenzyme. Also learn the clinical correlation. Now, if we learn decarboxylation, I should say something along with it. I will give you some examples here. Pyruvate is converted to acetyl coenzyme. Another one is alpha ketoglutarate is converted to succinyl coenzyme. The third example is branched chain keto acid. Branched chain keto acid is converted to its corresponding acyl coenzyme. In all this reaction, if this is a 3-carbon compound, this is a 2-carbon compound. This is a 5-carbon compound. This is a 4-carbon compound. This can be N number because there are various branched chain keto acids. So I'm just writing N. This is N minus 1, which says in all this reaction, carbon dioxide is removed. So is it belonging to lyase? No. Why? Because along with this, NAD plus is converted to NADH. NAD plus is converted to NADH. Here also, NAD plus is converted to NADH, which says that this is not a simple decarboxylation, it is an oxidation reduction reaction, because here it is reduction, so correspondingly the other reaction should be an oxidation, which says that this is not simple decarboxylation, this are oxidative decarboxylation. Okay, now to which class? First, let me name it. Now, these enzymes are called as, I'll give you two options. Is it decarboxylase, just like what we have learned now, or is it dehydrogenase? Whenever NAD2 NADH is there, we should not call it as a decarboxylase. We have to call it as a dehydrogenase, right? So, this is dehydrogenase. Okay, so I'm writing DH here. So correspondingly, all these are DH. What DH? Write the name of the substrate, pyruvate dehydrogenase, alpha ketoglutarate dehydrogenase. This is branched chain keto acid dehydrogenase. Now, all these are not simple decarboxylation. These are oxidative decarboxylation. Okay, fine. So what? Does it belong to class 4? No. This is not belonging to class 4. This is belonging to the class 1 oxido reductase, which is coming under the subclass dehydrogenase. Okay, so what is a coenzyme? In the case of simple decarboxylation, what was the coenzyme? B6. Now, what about this? There are 5 coenzymes for all these reactions. 5 coenzymes. One is thymine pyrophosphate. Second is coenzyme A. Third is lipoate or lipomide. Fourth is FAD. Fifth is NAD plus. All the 3 reactions that I've given above, PDH, BCKD, and alpha ketoglutarate dehydrogenase, all these require these 5 coenzymes. Very, very important. Then all these belong to class 1 oxidoreductases. And one more common thing that you can learn for all these enzymes which I have given here, whether it is PDH, whether this is alpha ketoglutarate dehydrogenase or BCKD, all these are multi enzyme complex. We will be learning this in metabolism as well. Okay, so these are some general thing always keep in mind, which will help you to remember things easier. Otherwise, we will have to learn everything separate. So that always make us things forget very easily. So if we group and learn like this, it is very easy. So what we have been discussing is about the class 4. Under that, we have learned about the simple example aldolase, then fumarase, enolase, then every enzyme with lyase in its name. Then we learned decarboxylases. Under that, only simple decarboxylation we can include. Then the oxidative decarboxylation is coming under class 1 enzymes. Okay. Now coming to the class 5, which is isomerase. We all know what are isomerization reaction. They are structural or spatial, or like the structural isomers where the molecular formula is the same, but they are different in its structural organization. They are enzymes which forms their corresponding structural or geometric isomers. So they are called as isomerase. There are 3 major subclasses. One is isomerase itself. Second is mutase. And the third one is racemase. So what are they? So the first one is isomerase. Example is glucose 6-phosphate is converted to fructose 6-phosphate. Then dihydroxy acetone phosphate is converted to glyceraldehyde 3-phosphate. Okay, so in all this reaction, what we can see is a 6 carbon is converted to a 6 carbon. A 3 carbon is converted to a 3 carbon. Okay. In all these reactions, it is like a formation or production of an isomer where the molecular formula, whether it is glucose 6-phosphate or fructose 6-phosphate, the number of like each of the atoms is same in both. Like the molecular formula is the same in either case, in the substrate as well as in the product side. But what is the difference is they are isomers, right? Okay. Now, in the case of glucose 6-phosphate to fructose 6-phosphate, it is like the enzyme is isomerase. Both are isomerase. Now, we have to prefix it with a name. That is, what isomerase is it? They are both phospho. So, I'm writing here phospho. This is a hexose. The fructose is also a hexose, right? So this is phosphohexose isomerase. Now what about this one? This is isomerase. This is a 3 carbon. That is a triose. This is also a 3 carbon triose. Now phospho is common in either side. So I am writing here phospho. Now these are triose. So I am writing triose here. So it is phosphotriose isomerase. So these are the two examples that is coming under isomerase. Now under the isomerase, I've told you 3 examples. One is the name isomerase. Then 2 more examples are there. The second one is mutase. Mutase is glucose 6-phosphate is converted to glucose 1-phosphate. One more example is 3-phosphoglycerate is converted to 2-phosphoglycerate. So in these reactions, what is happening? Glucose 6-phosphate has a phosphate group at the 6th position, whereas glucose 1-phosphate has a phosphate group at the 1st position. So it is like an intramolecular transfer of functional groups. Like in this case, what we can see is a 3-phospho. This is 2-phospho, which means in the first case, the phosphate group is at the 3rd position. In the second case, the phosphate group is at the 2nd position. So it is like an intramolecular transfer of phosphate. And these enzymes are called as mutase. What mutase? So this should be phospho both sides. So I'm writing phospho here. Then there is a gluco here. I'm writing phosphoglucomutase. Glucomutase. What about the other one? It is both are phosphoglycerate. So I'm writing phosphoglycerate mutase. Phosphoglycerate mutase. Okay. Now, what is the difference between mutase and kinase? So that I will explain it with a small demo clip. To explain the difference between kinase and mutase, we are going to do a small role play. So for today, you are taking the role of what? Creative. Yeah, he is taking the role of ATP. You know, ATP is having 3 phosphates. So you can see 3 phosphates here. Now, I am taking the role of glucose. How many carbon atoms are there in glucose? There are 6 carbon atoms. But I don't have 6 hands. So, I am just showing my one of my hand. This is the first carbon atom. And this is the 6th carbon atom. Okay. Now, what is a kinase? So, what is the action of a kinase? A kinase is an enzyme which transfer a phosphate. So, can I take this? So, this is a phosphate. I'm taking this phosphate to my 6th position. So what is the change that happened to me? I became from glucose to glucose 6-phosphate. So this action is done by an enzyme called as kinase. And remember, this belongs to the class 2, which is transferase. Okay. Now, I'm going to explain what is mutase. Mutase is like, now, can you name me? I am glucose 6 phosphate, right? So I am transferring this phosphate to the first position. Now this glucose 6 phosphate become glucose 1 phosphate. So which is the enzyme has which has done that function? It is a mutase enzyme. So when I transfer the phosphate from the 6th position to the first position, my molecular formula change or not? No, because whatever phosphate I have, now also I am having the same phosphate with me, which means my molecular formula is not changing. If my molecular formula is not changing, the reaction or this reaction that is happening here is an isomerization reaction. And again, this is a transfer of phosphate. So when I took phosphate from the ATP, now becomes glucose 6 phosphate. It was a transfer of phosphate from one substrate to another. That is not an isomerization reaction. But when I transfer the phosphate from the 6th position to the first position, my molecular formula is not changing. So this is an isomerization reaction, and this enzyme is a mutase. So this belongs to the class of isomerase. Okay. So this transfer is both are transfer of phosphate. So this is a transfer from one substrate to another. Now it is kinase. Now when I transfer it from one of my carbon atom to another carbon atom, which is called as intramolecular transfer of phosphate, this is not kinase. This is a mutase. So this is glucose 6 phosphate to glucose 1 phosphate. This is by an enzyme called as mutase. Hope you understood what is the difference between a kinase and a mutase. Thank you. So under isomerase, we have learned the same name isomerase. Then we have learned what are mutase. Then what we are learning is the racemase. It is very simple. We know that D and L isomerism. D and L isomerism. So any enzyme which create this D and L isomers, they are called as racemase. For example, D-alanine is converted to L-alanine. D-glucose is converted to L-glucose. So they are given the name racemase. Okay, so this is coming under isomerase. Now we are going to the 6th class of enzyme, which are called ligases. Ligases, as the picture says, that it is joining of two substrates, and especially in this case, we know that in this movie it was like fighting against the odds. These two substrates are joining, so it requires like lots of efforts and energy, right? So it was not a simple joining of two substrates, just like the story that you all know. So just like that, in the case of ligase, it is joining of two substrate with the involvement of hydrolysis of ATP, where the energy from the ATP is used. Okay. So it involves hydrolysis of ATP, and the two substrates are coupled together. So under this, we have two examples. One is synthetase. The second is carboxylase. Okay. So the first example under ligase is carboxylase. It's very interesting to learn about these three carboxylases together. That is pyruvate becoming oxaloacetate, then acetyl coenzyme A becoming malonyl coenzyme A. One more example is propionyl coenzyme A becoming methyl malonyl coenzyme. In all these reactions, what is common? This is a 3 carbon, this is a 4 carbon. This is 2 carbon, this is 3 carbon. This is 3 carbon, this is 4 carbon. Okay, fine. Now in all these reaction, it is like a addition of a carbon dioxide. So all these are carbon dioxide fixation. So that is why they are called as carboxylases. It is like adding of a carbon group extra there, so that 3 to 4, 2 to 3, like that. Now in all these reaction, 1 ATP is required because ligases require hydrolysis of ATP. It becomes ADP. And the second thing is that they require biotin. The third is that they all involves addition of a carbon dioxide. So I can take out 3 alphabets from here, A, B, C. And what is the name of this enzyme? It is a carboxylase. In this case, this carboxylase is pyruvate carboxylase. Now, in all the other examples, I'm just writing these 3 alphabets and you will be clear of that idea. All these enzymes require A, B, C. That A stands for ATP, B stands for biotin, and C stands for carbon dioxide. Here also A, B, C is involved. ATP, biotin, and carbon dioxide. And in all these, the enzyme's name is a carboxylase. Here also it is carboxylase. So what carboxylase? In this case, it is acetyl coenzyme carboxylase. In this case, it is propionyl coenzyme carboxylase. So what? All these carboxylases belongs to ligase and all these require A, B, C. So we usually call these three enzymes together as ABC enzymes. So all these are carboxylase. And what is the vitamin for all these carboxylation reaction? The vitamin for all these carboxylation reaction is biotin. So is there any biotin independent carboxylation? Yes. The biotin independent carboxylation are carbamyl phosphate synthetase 1 and 2. One carbamyl phosphate synthetase 1 is for urea cycle, 2 is for pyrimidine synthesis. Then gamma carboxylation. For gamma carboxylation, we require vitamin K. The third is malic enzyme. In the malic enzyme, it's also a biotin independent carboxylation. And the fourth example, one more is there. It is AIR carboxylase. AIR carboxylase, which is a reaction in purine, de novo purine synthesis. De novo purine synthesis. Okay, we will learn all these things in the corresponding metabolic pathways. I'm not explaining it here, but still we should understand these are the biotin independent carboxylation. So we have been learning about the ligase, where we learn about the carboxylase. The second subclass under ligase is the synthetase. So in the synthetase, it is like joining of two substrates where ATP is involved. The examples are all enzymes with synthetase in its name. So whenever we write the name synthetase in the name of the enzyme, always we can write the conversion of ATP to ADP, or sometimes it can be ATP to AMP. Anyway, what we should know is that it involves hydrolysis of ATP. So the examples of synthetases are 1-arginino succinate synthetase, then carbamoyl phosphate synthetase. So these are all examples of synthetases where ATP is required. Okay. So now we have reached the last class of enzyme, which is translocase, which is the recently added one in August 2018. It is involving the transfer of molecules and ions across the membrane. So most of the examples of translocases are calcium channel, H plus pump, potassium channel, etc. Fine. So these are all belonging to the class of translocase. So we have learned lots in this session, right? So we'll just summarize what we have learned. First thing what we have learned is there is a unique numbering system for every enzyme. Under that, there are 7 classes of enzymes. Some enzyme class, we learn the subclasses as well. So the first one is about the class 1, where we learn about the subclasses like dehydrogenases, oxidase, oxygenase, and hydroperoxidases. Dehydrogenases transfer the hydrogen to another substrate, which is usually a vitamin, that is a B complex vitamin like NAD, FAD, NADP plus, etc. In oxidases, these H plus elements are transferred not to a vitamin, but it is to another oxygen. So the example is cytochrome C oxidase, where we know that the final electron acceptor is the oxygen. Then oxygenase is directly adding oxygen into the substrate. There can be adding of one atom of oxygen or both atoms of oxygen. If it is one atom of oxygen, we call it as monooxygenase. If it is two atom, we call it as dioxygenase. Then hydroperoxidases, we have learned two examples under that, peroxidases and catalases. Both of them use hydrogen peroxide as a substrate, but the electron acceptor can be like glutathione, quinones, vitamin C, etc. Then it is called as peroxidase. If the electron acceptor is hydrogen peroxide itself, then it is a catalase. So under the catalase, there is only one enzyme, that is catalase itself. So remember, under transferases, kinase, phosphorylase. Then hydrolases, we have learned it is like a cleaving of a covalent bond by adding water. So under that, we have learned all digestive enzymes. Then phosphatase, we learn under that. Then lyases is like it's a tricky class where different names are there. One is just cutting of the covalent bond without adding water, aldolase. Then fumarase and enolase, where either atom elimination and forming a double bond or adding atom across the double bond so that a single bond is formed. So all these are examples of lyase. And decarboxylase also we learn. So under the decarboxylase, we learn the simple decarboxylation. Oxidative decarboxylation is coming under dehydrogenases, which is a class 1. Then comes the isomerase, where we have learned isomerase itself, where it is forming its structural isomer, where we have seen glucose 6-phosphate to fructose 6-phosphate, etc. Then we learn about mutase and racemase. Then ligases is joining of two substrates, where we have learned the example of carboxylase. 3 examples of carboxylase, they are all coming as like 3 alphabets are required for all these carboxylases, that is ABC. So this is very, very important. Then comes the synthetase. It is also a ligase. Then the last one is the translocase, where it is transferring of ions across the membrane like the potassium pump, calcium channels, etc. So this is the summary of all the classes of enzymes. So to conclude, if you cannot do great things, do small things in a great way. It's very important to have success in our life. We always think that successful person have done many things great. No, it's not like that. They have done small things in a great way. That makes them exceptional. So the same way, if we can do something, a small thing in a great way, that is something greater than doing something great in a great way. So that is not going to happen. We are all humans. We all have our own shortcomings. But in spite of that, we can succeed if we can do small things in a great way. Thank you.